Hello everyone. Today I'm going to explain about Griffith experiment. Let's start from an introduction. Frederick Griffith conducted a series of experiments with the Streptococcus pneumonia and pneumonia bacteria and mice in 1928 and concluded that the Ashton bacteria must have picked up a transforming principle from the heat-killed S bacteria, allowing them to transform into a smooth-coated bacteria and become virulent. A Griffith experiment carried out by the English bacteriologist Frederick Griffith in 1928. Frederick Griffith was an English bacteriologist. He conducted this experiment in 1928 and he described that the transformation of non-pathogenic like bacteria into a virulent strain. Okay, in this experiment he showed the transformation of non-pathogenic uh, pneumonial bacteria into a virulent strain. Okay, Griffith combined living non-virulent bacteria into a heat inactivated virulent from this experiment. Okay, let's get on to the experiment. He was the first to discover the transforming principle which led to the discovery of DNA as a carrier of. Okay, he was the one to first discover the transforming principle which led to the discovery of DNA. He conducted this experiment basically to find a cure for a pneumonia or vaccine for a pneumonia, but he ended up by discovering a DNA as a genetic material, genetic information carrier. Okay, he suggested bacteria can refer genetic information via was a process is called. Okay, this process, the bacteria can transfer genetic information. This process was known as the transformation. The Frederick Gift was not to identify the his main goal was not to identify the genetic material, but to create the vaccine against pneumonia. He mainly conducted this experiment to find a vaccine against the pneumonia which was the very deadly disease on that time. He mainly started fight to find the create a vaccine to against pneumonia, but he ended up by finding a genetic material. In this experiment, Griffith used two related strain of bacteria known as R and S strain. Griffith works was expanded by Arwen. Okay, in this experiment, Griffith used two strains of bacteria. Like we have many strains of bacteria, some strain, but he took two related strains, like a strain of bacteria and R strains of bacteria, and his works was expanded by Arve, McLeod, and MC Catty. Streptococcus pneumoniae comes in several types of strains. Griffith chooses two different strains of for this experiment. Already we know that he chooses the S strain and R strain. One strain of the bacteria has smooth surface and is known as a smooth strain, means S strain, while the other has the rough surface and is known as the rough strain or R strain. Okay, these were the two strains which he selected. Bacteria of the strain have the smooth surface because they produce an polysaccharide protective coating that forms an outermost layer. Okay, the strain layer is so smooth surface, so because of it produces a polysaccharide protective cavity. The strain is covered by a protective cavity which is formed by the polysaccharide. It's like a coating to the from the outermost layer. Apart from the morphological differences, Griff discovered another significant difference between S strain and R strain of bacteria. That is, S strain is a virulent strain and capable of causing death in a mice, whereas R strain is a non-virulent strain that will not cause death in the mice. Okay, what happened here? He took two strains, which is Ashland. The major morphological difference was one was having the polysaccharide coating, another was not having a polysaccharide coating, and another main morphological difference was A strain was a virulent strain and R strain was a non-virulent strain. The A strain, which is virulent, it was capable of causing a death in a mice, but R strain is a as is a non-virulent strain, it has a no capability of causing a death in a mice. Okay, Griffith observed that when he injected this bacteria into mice, the mice infected with the virulent strain died from pneumonia, whereas the mice infected with the non-virulent strain survived. Okay, we can see in this figure, when Griffith injected the strain bacteria to a mice died by it's a virulent. A strain is a virulent, but when he injected the R strain to the mice, it survived, mainly because here we can see in this figure that this A strain bacteria. Here we can see the lightish red coating. It's a polysaccharide coating which was coated to the S strain. It act as a like protective layer. Okay, this is R strand which has no outermost layer or protective layer or capsule something. So these are the two strands he selected. Okay, Griffith transformation experiment. Okay, Griffith was researching the possibility of developing an pneumonia vaccine. Okay, we already know his main goal was to develop an vaccine for the pneumonia. He used two strains of pneumococcus or streptococcus pneumonia bacteria that infect a mice, a virulent A strain and non-virulent type R strain. These were the two strains which is taken. The estrine produces a polysaccharide capsule that protects itself from the host immune system, resulting in the host death. And whereas Astran lacked that protective capsule and was defected by the host immune system. Okay, and last picture we have seen, this is the like outermost capsule, polysaccharide capsule, which act as a protective layer from the host immune system, whereas immune system can't penetrate to this layer, upper layer, so it protect the virulent inside this, and which helps causing diseases and in causing the death. Here we can see Astral has no capsule or protecting layer over it, and so it will be affected by the host immune system, which will be avoiding it from causing any diseases or causing any death. Okay, Griff attempted to inject a mice with a heat kill S bacteria as a part of his research. That is, S bacteria has been heated to high temperature, causing the cell to die. The heat killed S bacteria, but unsurprisingly, it did not cause any disease in the mouse. Okay, as a part of his disease, S strain was a virulent bacteria, so it causes the disease and death in a mice. So what he did after injecting a S bacteria, he took an S bacteria again and he heat killed the S bacteria strain, and he in a high temperature, and again he injected to the mice. But unsurprisingly, what happened, the mice didn't die or has been not affected by any disease when so it was unsurprising was. But when harmless R bacteria were combined with harmless heat killed S bacteria and injected to a mouse, the experiment took an unexpected turn. Not only did the mouse develop a pneumonia and die, but Griff discovered the S bacteria in a blood sample taken of the. Okay, what he did, he finally, okay, the mouse survived when he injected the heat kill S bacteria. Then what he did, he took the R bacteria strain and S bacteria strain, he combined it and he injected to the mice, and where he observed an unexpected and surprising turn. The mouse developed an pneumonia disease and died. Okay, beside of this, when Griffith uh, took a sample of a blood of this infected mice and go through it, he the living S bacteria was found in blood sample of the dead mouse. He concluded that some factor or biomolecule from the heat kill S bacteria had entered the living R bacteria, allowing them to synthesize a polysaccharide coating and become a virulent. As a result, this factor transformed the R bacteria into S bacteria. Okay, finally, after this going through the blood sample of the dead mice, he concluded that some factor or biomolecules from heat kill strain, which has been already heat killed, so heat has entered the living R bacteria. This heat killed S bacteria has no like, it has become already non-virulent or something, but when it enters the living R bacteria, it allows them to synthesize a polysaccharide coating. Here when it is heated, heat kill strain was, it was not coated, it was already like discoated or something, but when it enters the R bacteria, it synthesizes a polysaccharide coating and it becomes virulent. The mainly R bacteria was a non-virulent strain, but when this heat kill strain bacteria was entered to the R strain bacteria, it synthesizes the polysaccharide coating and it has become virulent. As a result, when it becomes virulent, as we know, it causes a disease in a mice and it it causes the death. And this factor they transform the R bacteria into the S bacteria. Griff called this factor the transforming principle, concluding that it carried some genetic material from the S strain to the R strain. Okay, the mainly like when a heat kill S bacteria enter the living R bacteria and it produces the polysaccharide coating and it becomes violent. This is mainly due to the transformation of genetic material from S bacteria to R bacteria, and this Griffith called this as a factor that transforming principle. This factor as the transforming principle. The process is now known as the bacterial transformation and is used in variety of significant genetic engineering application. Okay, now this process is mainly known as bacterial transformation, and it is significantly used in all the genetic engineering applications. It's many. Okay, here's the diagrammatic representation of this experiment. Here we can see the rough strain means R strain, which is a non-virulent. When it's get injected to the mice survives because it's a non-virulent, and it's a smooth strain which are virulent. Here we can see the coating, like orange, reddish orange coating, polysaccharide coating, which has act as a protecting layer or outermost layer of the strain. And when it's get injected to the mice dies. And here comes the heat kill estrine. Here when is heat kill estrine is injected to the mice, the but mice survives because it is heat killed already. But when rough strain means R strain and heat kill estrine are combined together and injected to the mice, here we can see we have both estrine means heat kill estrine, and here we have rough strain, it is combined, it injected to the mice, but mice dies because it when estrine enters to the astrine, it makes the astrine to develop an polysaccharide coating and it becomes violent, which causes the death in the mice. Okay, one of the like in fact one of the characteristics of hereditary material is a changing phenotype. Okay, a changing phenotype is a characteristic of this hereditary material, and Griff referred to the phenotypic changing factor as a transforming principle. Okay, his work on the transforming principle received the most attention, but only after a group of Canadian and American scientists set out to investigate the chemical nature of the transforming principle in Oswald Harvey's laboratory. Okay, in his transforming principle received the most attention on that time because it was a like new discovery. So he has a more attention on his principles and transforming principles, but only after a scientist of Canadian and American set an investigation the chemical nature, the transforming principle, which is the chemical nature of the transforming principle. It was held in investigation was held in the Oswald Harvey's laboratory. Harvey's group concluded in their studies that deoxyribonucleic acid was the molecule identified the Griffith as the transforming principle after conducting numerous experiments. Okay, after conducting investigation or something, after going through under numerous experiment, the Harvey's group concluded that DNA means deoxyribonucleic acid was the molecule identified by the Griffith as a transforming principle. Okay, the implication of this discovery are far-fetched, far-fetched because it was made at a time when scientists consider protein molecule to be the genetic material. DNA or deoxyribonucleic acid is now recognized as the molecule that encodes all cell functions and transmits genetic information from parent to offspring in almost every living species. Okay, this what DNA as a genetic material was found in that time where the all the people were considered that the protein was the molecule to be the genetic material. So when he discovered that DNA was a like transforming a genetic material, people were like it was far-fetched, it was like unimaginable, or they were not ready to accept firmly. So they all considered protein molecule to be the genetic material at that time. But then now DNA is recognized as the molecule that encodes the cell function and transmit genetic information from parent to offspring in all every living species. Okay, in 1940s, however, DNA was thought to be a less qualified candidate for genetic material. Irvine and Scola research on Griffith experiment provide the first solid evidence that DNA could be the genetic material. Okay, however, we know this all on at that time the scientists consider the protein as the molecule to be genetic material. So when the Griffith find out that DNA was an genetic material, they it was a less qualified candidate DNA, because all were considering that the protein was the genetic material, so they were not ready to accept that DNA was an genetic material. So DNA was a less qualified candidate of for the genetic material. But after Irving and his colleagues researched the Griffith experiment and provided the first solid evidence, it was the Irving and colleagues research was the first evidence, like solid evidence for the DNA could be the genetic material, not protein. Okay, conclusion of the Griffith experiment. Griffith's ultimate goal was to find a way to the cure pneumonia. Griffith inoculated mice with the various strain of pneumococci to see if they would infect and eventually kill the mice. Griffith concluded that heat killed virulent bacteria transferred living non-virulent bacteria into virulent bacteria. He performed his experiment on the two strains of Streptococcus pneumonia, which differ from each other due to the presence of polysaccharide growth. Okay, the Griffith main intention was to find the cure for the pneumonia or vaccine for the pneumonia, but when he injected the various strain to the mice, and it got infected, eventually killed the mice. Like a heat killed virulent bacteria transferred leaving to the non-virulent bacteria into the virulent bacteria. Heat killed virulent bacteria transferred to the living, and it becomes non-virulent to the virulent. It was because of the transformation of genetic material. And this experiment was our two strains. Already we know the two strains were the S strain and the R strain from the Streptococcus pneumonia, and which were differ in the just in the protein coat, polysaccharide protein like coatings. Okay, Griffith findings were published in the Journal of Hygiene. In 1928, his experiment worm mice led to his major discovery of bacterial transformation. Griffith's experiment discovered that bacteria can transfer genetic information through transformation. Okay. Conclusion of this experiment was that he discovered that bacteria can transfer the genetic material, genetic information from through transformation. Okay, this was all about the Griffith experiment, and thank you.